Hello and welcome. We're going to spend a few minutes today talking about the basics of how local anaesthetic actually affects neurons. Now, to set the context, remember that for pain information to flow back, this is a neuron I'm drawing here, from a painful stimuli. Let's draw ourselves a splinter or something nasty here. From a pain stimuli at one end back to the brain. Remember, it can be via the spinal cord, obviously, but for that information to travel back down the neuron, it travels as an action potential, which is a little sort of pass the parcel electrical Morse code that runs down the neuron. Now, just out of interest, it can run at relatively high or low speeds, and just think for a minute about how slowly pain information actually does travel down neurons. Have you ever noticed that when you pick up a hot plate, that you're staring at it and you can see that it's hot, but you don't actually feel the pain until a wee bit later? And that's because the information travelling down the pain pathway travels relatively slowly. So the key to this information flow down a neuron is the transmission of action potentials. And just as a reminder, what is an action potential? Well, an action potential is where normally the inside of a neuron is a particular negative charge and the outside is normally positive. I'm going to run out of oomph drawing all these, but we'll just draw a few in for now. There we go. And what happens when an action potential is in place is that this flips. And then that little patch returns to normal, and then further on the flip happens, and then behind it returns to normal. So we'll just colour this in, say that all disappears and returns to normal back there. And then this flipping of positive and negative stimulates a little bit further down the track for it to flip, and at the same time behind it returns to normal. And slowly you can see this electrical flip slowly moves down the neuron. And that's how information is passed down neurons. So the question now is, how does local anaesthetic work? Well, the obvious thing that you want to do with local anaesthetic is that you want to block the flow of information. So you want to stop the action potentials from getting back to the brain, because it's back at the brain that you actually register in your mind and feel the pain. So we want to stop the information getting back there. And to do that, we want to stop these action potentials. So they come up to this point and they can't go any further. And that's what local anaesthetic does. Local anaesthetic blocks the transmission of action potentials down a cell membrane. So let's talk about how that happens. Let's draw ourselves just one piece of cell membrane, and please remember, you need to understand how cell membrane works to understand this process, and there is a separate video that will explain to you how cell membrane normally works. So like I said, cell membrane on one side has one charge, this is of a neuron, and on the other side is normally another charge. The opposite, obviously, negative and positive. And the way this happens is that the cell membrane of a neuron has embedded in it, and I'm drawing it huge, just imagine it's all within this blue line here, has embedded in it a protein that pumps sodium and potassium ions in opposite directions. And this sodium, potassium, let's draw that again. Sodium, potassium pump keeps the membrane at a potential, or keeps the positive on one side and the negative on the other. So you can now work out exactly how the simple answer to local anaesthetic works. If you can block this pump somehow, you can stop the local anaesthetic. You can stop the flipping of the charges, and therefore you can stop action potentials. So local anaesthetic blocks this pump. So LA is a blocker of this pump. But it's not as simple as that, because the pump is quite unique. The only place you can control this pump is actually from the inside, from the cytoplasm side. Let's just write this so we make sure we remember this is the cytoplasm side. So you can only block the pump from the cytoplasmic side. So this means that when you inject the local anaesthetic solution, let's imagine you inject a great big blob of it here, this local anaesthetic solution has to somehow find its way through the cell membrane to then attach itself to the pump to block its action, and that will then stop action potentials running down this cell membrane. But we have a little bit of a problem, and if you remember back to our discussion of cell membrane is made of fat. Whereas the intercellular fluid is a watery environment, as is the cytoplasm. So the problem we have is that the local anaesthetic has to be water soluble, so you can inject it on the outside of cells. It has to be water soluble to be in the cytoplasm to then move around and do things. But for just an instant, as it tries to pass through the cell membrane, it has to be fat soluble, because otherwise it can't get through this fatty wall that makes up the cell membrane. So this is true. Local anaesthetic molecules are actually a very cunning chemical process, and their chemical process allows them to be in a mixed state of water soluble and fat soluble. And it's the fat soluble that can then rush through the membrane, and then it can split back into its sort of two-state life, and the water soluble can then do the blocking of the sodium potassium pump. So let's draw that out and explain how that happens. So here's our cell membrane. And what I'm going to do is I'm going to draw 1,000 molecules of local anaesthetic. Well, I'm not going to draw 1,000, I'm just going to write, let's just write 1,000 here. 1,000 LA. Now that's not actually how local anaesthetic is. What actually happens is it balances out to be about, and it depends on the solution, depends on all sorts of things, so we're not going to worry about the actual numbers, it's only for demonstration purposes, these numbers. About 800 of the molecules would be water soluble, and 200, and I'm going to use yellow for fat. In fact, I probably should have drawn the cell membrane as yellow. Let's just draw in some yellow to remind us that the cell membrane is made of fat. There we go. And so this is sort of a balanced equation of how the local anesthetic molecules sit in the water. About 800 are water soluble and 200 are fat soluble. And clearly we've got a solution here. We've got a fantastic answer, because these 200 fat soluble can move their way straight through the cell membrane. And then what happens is the 200 break up into the same sort of balanced 8-to-2 or 4-to-1 mix. So we have, I have to work it out now. I don't know the maths, but it'll be the same sort of 4-to-1 ratio. Let's say it's, it'll be about 160, won't it? 160 to, actually, let's just write that again because we'll get the colours exactly right. What colour are we doing for water soluble? We're doing pink, aren't we? So that should be, let's just cross that out. We'll write that again. 160, and there'll be still fat soluble will be 40. And then the good news is that those 160 water soluble molecules of the local anesthetic are now available to block the sodium-potassium pump. So this is interesting, isn't it? Because if you think about this, you injected 1,000 molecules, but really, in the way the chemistry worked, only 160 were actually available to effect the critical step in blocking the action potentials. Because remember, that's what this is about. When you block this gateway here, this sodium-potassium pump, the action potentials are stopped. They can no longer go past this point. So the chemistry of local anesthetic, being able to mix between water and fat soluble, is a critical part to allowing it to be effective. And the ratio, see in this 800 to 200, the ratio is really critical in telling you how effective the local anesthetic is. Because let's just imagine for a minute that this was 900 to 100. You would only have 100 to cross the line here, so this would only leave you very few then water-soluble ones to affect the sodium-potassium pumps. So the ratio between water and fat soluble is a critical part to understanding a local anesthetic molecule. Now, for now, the chemistry is too complicated. We're keeping this really simple. But just to highlight, if you ever see written pKa, you will know that this is the chemical measure of the ratio between the water and fat soluble phases of the local anesthetic molecule. So just to summarise, local anesthetic works by blocking the sodium-potassium pump. Sodium-potassium pump. And if you're not into chemistry, just to remind you, Na is the symbol for sodium, and K, do it nicely, is the symbol for potassium, sodium-potassium pump. So local anesthetic works by blocking the sodium-potassium pump that stops action potentials from heading back towards the brain. And the way it can only do that is from blocking those pumps from within the cell, so within the cytoplasm. And to get into the cytoplasm, it has to make its way through the fatty cell membrane. And to do that, it has two phases, a water and fat soluble phase simultaneously. And the fat soluble phase can dash through the cell membrane to then break back into its two phases, fat and water soluble, which is really good news because it's the water soluble component that can sit against the protein that is the sodium-potassium pump and block it. And that's how local anaesthetic works. Thank you.